Good afternoon, and thank you for joining us for this afternoon's RSSL webinar on cleaning validation. We're just going to take a few minutes to let a few more people dial in, and then we'll start, so bear with us. Thank you for waiting. Now that everybody's joined us, we'll start today's session. This is the first of 3 sessions that we are running on this topic. Today's webinar will focus on the importance of a good plant inspection to a successful cleaning validation outcome. Brian Hammond will be delivering the webinar this afternoon, and I will be here to support him. My name is Lisa Hallwood, and I'm a Senior Account Manager here in our commercial team at RSSL. Brian Hammond is one of our technical consultants within our training and consultancy team at RSSL and has many years' experience in supporting our clients with their cleaning validation needs. So before we get started, let's just share with you some information in case you're having any technical issues or also want to pose a question. For any technical issues that you may be having, please send a short message within the chat box on the right-hand side of your screen. The team here will then respond to you in order to try to resolve your issue. Whilst Brian is presenting, if you have any questions to ask, please feel free to submit these using the questions box on the right-hand side of your screen. We will do our best to answer these questions at the end of the formal presentation. Okay, so now that we've got the admin aspects out of the way, let's proceed with the webinar, and let me tell you briefly a little bit more about RSSL so you can understand why we are able to share with you our knowledge today. So RSSL was established in 1987 and has been supporting a number of industries, including food, healthcare and pharmaceuticals on a global basis during this period. We currently work for 20 of the top global companies as well as small startups and SMEs. Our FDA and MHRA inspected facility in Reading is where our team is based, and our technical expertise covers microbiology, chemistry, physical sciences and biochemistry. RSSL have been working in the pharmaceutical sector for more than 25 years, and our multidisciplinary teams support our customers from early phase development all the way through to finished product QC analysis. For some of our clients, we undertake all of their product development and testing. For others, they would use us for specific methodology that they either don't have the expertise or the equipment to do internally. Our emergency response service ensures priority access to laboratories 24/7 to support projects that are time critical, such as resolving customer complaints or manufacturing issues. In supporting the pharmaceutical industry, RSSL has a breadth of capabilities and a wealth of experience across a number of different areas, such as sterols or topicals, all the way through to oral dosage forms. We support customers with their own methodology, and we regularly work to pharmacopoeial monographs, such as the USP and EP, which are the most commonly used, through to others, such as the Chinese, Japanese, and Brazilian pharmacopoeias, which are being more frequently requested as our customers move into different geographical regions worldwide. RSSL like to partner with our customers, better understanding their needs and refining the project scope to keep things as simple as possible whilst ensuring that the regulatory requirements be met. This is especially relevant to the approach we take with our cleaning validation projects. I'll now hand over to Brian, who will present the technical content of this webinar and tell us more about the importance of a good plant inspection to a successful cleaning validation outcome.
Thank you, Lisa, and good afternoon and welcome. What I want to do today is just give you some idea of some of the areas that you should consider looking at to get a successful cleaning validation exercise. It is important that you realise that cleaning validation has been a legal requirement on pharmaceutical companies since 1992. And therefore you are vulnerable to being inspected on this by both the MHRA and by the FDA and other regulatory authorities. What we would like to look at today is the importance of a thorough plant inspection, why that is important and what you can do to make that as thorough as possible. Understand the factors leading to cleaning validation failures, what actually does cause the failures, and what can you do to avoid those. Understanding cross contamination, what sort of parts of the plant do lead to cross contamination, and what you can do to avoid that, and how do you get a good cleaning validation protocol. You should also remember the definition is documented evidence, and therefore your cleaning validation protocol is one of the most important of all the documents that you will produce as part of that validation. So first of all, understanding the process. It's very important that you understand your plant, you understand what you need to do, you understand where any contamination could occur, and you do something about that. Either that you try and engineer it out at the beginning before you run your process, so before you contaminate the plant, or you include it. If you don't include it in your cleaning validation and in your cleaning protocol, then it's likely to get missed, and that can lead to cross contamination and eventually analytical failures on that validation exercise. So you need a complete understanding of your plant. This is best obviously done, if you can, in the design stage when you're designing the plant for that process. It's no good trying to look at and do a decent plant inspection after the plant has already been contaminated. It should be done before possible, so that you understand and can engineer out any of the areas that you do find during that walk round, during that plant inspection. You need to know that plant very backwards, every pipe, nook and cranny of it, to know where contamination could occur and what you can do to avoid that. You need to look at your cleaning methods, not just the in-process cleaning, but the manual cleaning. How much manual cleaning do you do? Can you avoid manual cleaning? Are there ways you can redesign the plant or alter the plant round to avoid manual cleaning? Because manual cleaning is a big issue. It is difficult to control human beings, and getting two people to clean the plant to the same extent. It's like people washing their cars. If you get three people in the same car, and you'll get three different washed cars afterwards, depending on how good they are at cleaning it, or how much they like their car. So manual cleaning is very difficult. It needs to be heavily proceduralized. It needs to be in very great detail, so that you say exactly where you're going to, how you're going to clean it, how much solvent you're going to use, how many cloths you're going to use, what direction, and how long you take to wash it. So if you can avoid manual cleaning at all, then it is far better in the long run to avoid it. So things like if you've got a pan filter on a different floor and you have to pump it up to the pan filter, and then you then have to manually clean the pan filter. You've got two issues there. You've got a manual cleaning issue and you've got a pump, and pumps are notorious for holding back contamination. Can you move the pan filter down and do away with the pump and just gravity feed? Then you can clean in place rather than, it's a fairly minor alteration to the plant which won't affect the process, but has a big impact on the cleaning. You've suddenly lost the need to clean a pump, which is difficult to clean. You've have the need to you don't have the need for a detailed manual procedure to clean the pan filter. So there are things like that you can do. Can you move valves around on service lines to avoid areas of dead legs? Can you take dead legs out or obsolete equipment out and avoid having them in place? Because why contaminate something that you're not using? If you're not using it, then the bottom line is don't contaminate it in the first place, because you then don't have to clean it. If you use spray balls only to wash the plant out at the end, but you don't use them for the chemical process, then have them taken out for that. The engineering time to take them out is a fraction of the cost of cleaning them and analytically testing them at the end. Some calculations have been done on this, and it might cost you £100 to take out the spray ball and put it back at the right point, and it will probably cost you £4,000 or £5,000 to test it. So there is big financial paybacks from removing stuff at the beginning if you don't need it. Samplers is another one. If you're not using a microbore sampler because you're not taking in-process checks, then don't leave it in to be contaminated in the first place. Take it out and blank off the pulp for it before you start. Then you haven't got to clean it at the end. One good point is to look at process investigations. Process investigations can give you a lot of information about what might go wrong on that plant at some point, because it's happened in the past. So if you look back at all your process investigations and say, well, what actually caused this? What was the answer to the process investigation? Is that something that I've got to think about? I've seen material stuck in dead legs up in the roofs of buildings that went through to 3 processes further on before it contaminated the process, because the reason it was up in the roof was that it had been blown up by somebody who overheated a vessel, and then at a later date they happened to use a condenser and use this pipe in the roof, and it put it back down again onto a process three products later. So process investigations in that situation can tell you that there is a possible issue there, and you can look at it and include it in your validation to avoid a problem in the future. Make sure you fully train your staff, particularly in manual cleaning, that they understand why they need to all do it exactly the same way in great detail. Probably include the best way of writing your manual cleaning procedure is to get the staff to write it for you. I ask them all, when you clean this particular piece of equipment, how do you do it? And then combine them all together for the best result. And then everybody feels they have a buy-in, and they're more likely to do it reproducibly from there on. Because manual cleaning can lead to all sorts of errors. One person will not clean it as well as somebody else, and then you get cross-contamination and you get swab failures at the end. And they're probably nothing to do with the cleanliness of most of the plant. It's just that the operator didn't carry out that exercise as thoroughly as another operator had done. Swab training is also another one. We'll look at that later on. That your swabs at the end are what is going to be analysed, the analysis will tell you what's on the swab, not what's on the plant. So if what's on the swab is not representative of what is actually on the plant, then the result that you will get from the analytical at the end is meaningless. And swab training is very difficult. Swab training is a skill that you need to learn by repetitive testing of yourself, and there are techniques to do that. We'll perhaps talk about that a bit later on. Understand the errors that are in the testing. Why are swabs such a problem? What does lead to swab contamination? What does lead to failures? Because it's not necessarily the cleanliness of the plant that's involved. It can be the handling of the swab itself. It can be the storage of the swab materials, the source of the solvent that you use to swab, the skill of the operator. There are so many possible errors in swab testing that you have to make sure you eliminate all of them. Otherwise, you will never ever get a satisfactory result at the end. So good design and validation of the analytical methodology. We're going to talk about that more in the next webcam, but the one point I would like to make at the moment is that the analytical validation is time consuming. It can take up to 10 to 12 weeks. And this needs to be factored in on your timescales. It is no good going into a cleaning exercise without having validated analytical methods in place. It is a legal requirement to run all of your validation analysis on validated analytical methods. And I've come across a number of issues where people have said, oh, well, we have to start up processing on this particular date. And then you find they haven't done the analytical validation and they haven't got 12 weeks between now and that date to do it. And therefore they can't validate the process. So you need to tie that in. You need to put that time scale into the process and make sure that it's part of your whole overall plan for your validation. So what is the definition? The legal definition in EU law and in UK law is clean validation is documented evidence. So you need a full set of documents in full detail right through from your process design and your cleaning validation protocol, your master plan, your protocols, your outcomes, right through to your analytical documentation and all of your analytical validation. And unless all that documentation is there, then you don't have a successful cleaning validation. So it is documented evidence, and it's that documentation will provide equipment which is suitable for processing medicinal products. Several large pharmaceutical companies over the last 2 years have been very severely reprimanded by the drug authorities for this particular work. One company had all their products removed from the marketplace because their documentation couldn't prove that they were cross-contaminated. The drug authorities didn't say they were contaminated. They said the company couldn't prove they weren't, and therefore they didn't have the documented evidence to prove that. And the result of that was an extremely large problem for that company, both financially and in terms of their market share. So this definition is very important, that you have to have very good documented evidence that will provide that surety. Not might do, not may do once, but not the next time, but will do every single time you run it, whether you test it or whether you don't. Acceptance criteria. Up until a few years ago, acceptance criteria were calculated on the basis of batch size or therapeutic dosage, or very, very occasionally in terms of lethal dose 50. Well, we're not actually in an industry where we want to kill 50% of our patients to get the last data, so that's one that doesn't normally occur. The drug authorities now require you, as of the last 3 or 4 years, the drug authorities now require you to also obtain your acceptance criteria by calculating using toxicological data. This has proved to be somewhat problematical to smaller companies because they don't have access easily to that data. We have approached the drug authorities on this and got their viewpoint at a symposium about a few months ago, and their stance at the moment is that that data should be available to you if you need to, if you search for it enough, that the original registers of that product should be able to give you that information, which will then allow you to do that calculation. Their bottom line, the drug authority's bottom line, was that if you cannot get that data from the original manufacturers, then you should employ your own toxicologists to obtain that data for you, but they will not accept that you cannot do that calculation. The overall opinion at the moment with those people that have done those calculations is that normally the toxicological data gives you a higher figure, a higher acceptance level. Now, this is obviously good from an analytical point of view because it's easier then to not to have to go down to such low levels, but you may wish to use that level, or you may wish to just calculate that level but use the level from therapeutic dose, which is very often lower if you wish to be more on the safe side. That's purely an industry decision, but you must calculate it by the toxicological data, even if you don't then use that data to think. Some companies are taking it that they use the therapeutic dose as a trigger to do something, and they use the if it goes below the toxicological data, then that is an alert level, and but they actually use the lower level to monitor, and that's one approach that you could take. Plant inspections. These are often not properly conducted. In my experience, most of them are not done with the right team or the right amount of time, and they're not considered to give you much information. In fact, that is totally untrue. They give you probably one of the best pieces of information you can get. You need to investigate invest in expertise for these. You need a good team, which suggest 4 people. That would include a validation coordinator who will lead that plant walk round, somebody from the production side who understands the chemistry and understands the plant and itself and how that plant operates, an engineer to advise you how easy it would be to dismantle certain things, because it's no good putting something on your cleaning protocol saying we want to take that apart only to find that you need a crane to do it with, and that's not going to be possible. So an engineer is a very key person on this walk round, and you need an analytical. And one of the questions I often somebody from analytical I often get asked is, why do we need an analyst on a plant walk round? You do, because the analytical people are going to give you your final pass or fail. They have up to 12 weeks work to do. They know their systems and how many samples they can handle in what sort of time scales, and therefore they need to buy into the whole process. And they can advise you, well, we can't take 10 swabs of that area because we can't handle 10 swabs in the time frame that we have to do this analysis, and that time frame may be instrument dependent or it might be stability dependent. So a team of 4 is ideal. On a fairly simple plant, that walk round should take probably 3 or 4 hours. On a complex plant, it should probably take all day. So it does involve a lot of time, so you need to allow sufficient time to do the plant walk round, to talk to staff to find out how they clean certain areas and where they see. Very often the production people are the people that have the expertise as to where bits of the plant are difficult to clean and may hold contamination. You need that information so that you can design your cleaning process correctly. So, and if you have a really good understanding of the process and where it is and what you can do in an engineering basis to take things out, then your plant walk round will actually write your cleaning protocol for you. So it's one of your documents fully written in place. So the sort of things that can lead to validation failures are inadequate pre-planning and design. Things like not thinking about the analytical validation and then suddenly finding when you get near the end that the analytical validation isn't complete and you can't do your cleaning validation. Swab training can very easily fail your whole validation. If your swab training isn't good enough, then and reproducible enough and correct every single time, then the results you will get are only, as I said earlier, they're only what is on the swab and not what is on the plant. You have to be 100% certain that what is on the swab is representative of what is on the plant, because if it isn't, then you are completely wasting your time in doing the whole validation. Not allocating the swab points correctly, picking the easiest places to swab rather than where the most likely point of contamination is going to be. Say on a vessel, for instance, it's no good swabbing an easily accessible wall that you can see, and you can see whether it's clean and everything when you're doing a visual inspection, and swabbing the top of the vessel right up by the stirrer at the top, which you can't see, and stuff splashes up. And if that isn't cleaned properly, that can then produce contamination in the next process. So part of your plant walk round should be to write your swab point allocation. So it's one part of your cleaning protocol. Is a list of where all the swab points are and that you're happy with them. Discuss all the swab points and make sure they are the best places. It's not the bottom of a product tray, it's the corner is the best place. So make sure you have chosen all the best places and they are properly documented, and your swab people understand exactly where they need to take those swabs and they don't take them somewhere different. Failure to determine your time scales. We talked about pre-planning. It's obviously important that you understand all the timescales involved. How long is it going to take to clean the plant? How long is it going to take to swab the plant? How long is it going to take to do the analytical testing? How long is the analytical validation going to take? All of those timescales need to be included in your plan and your scheme. One of the things that you're legally required to do by EU law is to say right up front, right at the beginning, before you've done anything in your cleaning validation master plan, you have to state how long that plant is going to stay dirty before you clean it and test it. because if you and then you must stick to that time scale every time you clean that plant, because if you don't, you have only validated that plant as being cleaned from a dirty situation in that time frame. You haven't validated what would happen if you left it dirty for longer and stuff started to degrade on that plant. So you must stay to that timescale, and you have to say up front in your master plan that this is the timescale that you are going to adhere to. Make sure you've got all the right documents in place. You've checked them all, they're all fully there, and that includes things obviously like training records. I've seen whole validations fall down because one person didn't have in their training record one of the small jobs they'd done as part of the validation, and the whole validation collapsed because of it. And a large amount of money and work was wasted because of that. Make sure you've looked at any investigations on that plant, that as back as far as you can possibly go, to see if any of those things that have happened in the past could happen again, and if they did happen again, have you covered them in your validation documentation? Have you built something in that would cover that situation if it did happen? We hope that plant processes will run correctly every time, but they don't. Things will go wrong, and vessels will be overheated, and material will be thrown into pipes that it isn't normally in. Make sure that your process, if there's a situation like that, you've covered it. I've seen large amounts, up to 2 kilos of material, sitting in dead ends up in roof spaces, and it's sat there for months after months and process after process before it's finally been washed back down and contaminated, a much later process, because the condenser that used that line wasn't used for the processes in between. And when it was finally used, 3 processes later, it brought all 2 kilos of material down into that product. Make sure that you've got a plan at the beginning that lists everything that you need to complete that validation. What documents do you need? What training do you need? What do you need? What engineering work do you need? Everything that you need should be planned at the beginning, and then at the end, towards the end of the validation, you can check that plan back to say, yes, I've covered that, I've covered that, I've covered every attitude of this. So there's a few things that can cause it. Now, in terms of actual plant areas that can cause contamination, anything that has crevices, corners, dead ends can do it. Particularly dead ends. Obsolete plant that's never used but is still connected to the plant that you use can end up being contaminated, and therefore it's far better in your planning stage to remove that old obsolete plant. Shorten dead ends so they're as short as possible and don't hold large amounts of material. Service lines, make sure you've got all the right controls in terms of filters and non-return valves on all your service lines, and that those controls are as close to the point of contamination as possible, because service lines are 1/4 inch, and once they're contaminated you can't clean them and you can't test them. And it's surprising how much material you can put down a 4-meter long line. I've seen recalls from around the world of products that were caused solely by material sucked back down vac lines and then pumped back at a later date into another product. Once went to one factory where they had a central vac system monitoring the vacuum on 6 buildings, and they had no controls to stop the materials going back down the vac lines. And each building made a separate product, and when we checked the vac pump in the separate building outside. It had all 6 products in it, all of which could have gone back into any others of those. So make sure that those systems are in place and that they're in the right place in the lines, and if they're not, then move them to where they are correct. Things like, as I've said, condenser reflux lines, because if material gets bumped up there by accident, it will stay up in the roof space until you next use the condenser, and then it will come back down again. Anything that has a screw mechanism, particularly the underside of the threads and the screw mechanism, because you can't see the underside very easily, so you can't visually inspect it to see if it is clean or not. Lines that slope uphill or downhill, or change the angle at which they slope. There was a situation where it was actually in the food industry where a single line was changed between 2 vessels, and they got cross-contamination of one food product with another, and they'd never had it before. And that was purely by the angle of the line. The old line, the wash solvent bounced down the line and cleaned it properly. The new one was at a different angle, and it ran down one side of the line and didn't clean the other side. So again, understand your plant, watch plant changes. If you make any plant changes after validation, make sure they're covered by change control procedures, because you can ruin your whole cleaning validation after you've validated by making a minor plant change, like changing a pipe or changing a pump. Watch filter meshes. Filter meshes are designed to hold material. That's their purpose. They hold back solids, but they can also hold back contamination, and they are difficult to clean. Valves are very difficult to clean, and valves can take material back into the housings, and it can sit there for month after month, and then it can finally end up going back through the seals and back into the process again. So as part of your validation, you will probably need to strip down a number of valves totally and test them, not necessarily on a permanent run basis, but certainly on a validation basis. Sampling equipment is microbore. If you don't need to take in-process checks, then don't leave the sampling equipment in, because it will cost you £100 or something to take it out at the right point. It will cost you thousands to analyze it. Seals and gaskets, because of the nature of the material, they hold product and are difficult to clean. Recoveries in swab testing from gaskets is always much lower than glass or stainless steel. You need to know what those recoveries are so that you know where I'm there. So they're all the sort of areas that you would want to look at during a good, thorough plant walk round. And from those, you would design plant changes, anything you want to change, anything you want to take apart, where you're going to take all your swabs, and that writes your cleaning protocol. So basically, the generation of your cleaning protocol will take place at that plant walk round, or should do. So you'll have the engineer there, you will get agreement on dismantling and inspecting. You will have the analytical people will say, yes, we can handle that number of samples from that number of places. So you've written your swab plan and your testing plans. Decide what pieces of equipment have to be manually cleaned. Try and avoid that as much as possible, and redesign your plant if you can to avoid manual cleaning. But also make sure that if you have to do manual cleaning, it is properly, very heavily documented. Check your production records back, investigations, anything that has gone wrong. It's very easy to generate a cleaning exercise if the plant worked correctly every time, but minor problems in processing can cause major problems in validation, including validation. And finally, document all of your things thoroughly, completely, and make sure that you've done everything you need to do to meet the definition of cleaning validation, which is documented evidence that your procedures will every time produce materials that are medically safe and safe to the patient. Thank you very much. And over to Lisa.
Now that we've got my mic working. Thank you, Brian. That was a lovely overview that you shared with us this afternoon. Thank you also to the attendees who asked questions throughout. What we're going to do now is start working through some of those questions for you. Brian, we had one come quite early on during the presentation from somebody, and you may have covered it off later with some of your slides, but could you recap for us when in the whole cleaning validation process should the analytical component be considered?
Ideally, during the design stage, right at the very beginning, before you ever contaminate the plant. If you don't do it then, the analytical people could have up to 12 weeks work to do to get their methods validated. And they are not allowed by law to run samples of from a validation exercise without validated analytical methods. The law is quite clear about that, the analytical methods must be validated. And you need to build in the time, the 12 weeks. Obviously, your analytical people can give you some advice as to how many weeks they think it will take. It varies from one exercise to another. But you need to make sure that that time is there. It's no good saying, oh, well, I've just cleaned my plant, I'm ready to do the analytical, and I need to do it in the next 2 or 3 days when they have got 12 weeks work to do in between. So basically make sure you understand that that work is needed by law and that that has to be part of your overall plan, and it should be at the beginning.
Okay, thanks for that. Another question that came through was, what is your view upon the use of toxicological data to calculate acceptance criteria?
Thanks, Lisa. Yeah, this is a hot topic at the moment, and it keeps coming up during training sessions and during consultancy. It is a problem to a lot of the smaller companies because that data isn't easily available to them. I think, as I said in the presentation, we have talked to the MHRA about that, and their viewpoint at this point in time is that they would expect you to have had done that calculation, and they will not accept that you can't get the data to do that calculation. Their response to our question on this was that data is available in the original registrations, and therefore if you go back to the originating companies, you should be able to obtain that data. If you find that is not possible, then you should budget in to hire your own independent toxicologists to create the data for you, but they will not accept that you cannot calculate it that way. Whether you then choose to use that calculation is up to you, because the law says it's up to you what level you choose. So if you have 2 levels and the toxicological one is high and your therapeutic dose is lower and you have an analytical method that will meet the lower one, there is no real reason why you shouldn't go for the lower one. But at least you can show an inspector that you have done it by toxicological calculation. What you can't do is just say, oh, I can't find that data and we won't do it. That is not acceptable to the regulatory authorities.
Okay, one last question for today then. How in-depth should swab training be?
Very in-depth. It is probably 50 to 80% of your failures. We investigated this very heavily about 30 years ago, when we were initially looking at cleaning validation back in the late 1980s. And because people were washing and cleaning plant multiple times and still getting failures and couldn't understand why. They couldn't understand why visual inspections and everything else and the washes and the rinse samples and everything were all clean, but the swabs were failing. It is extremely easy to contaminate a swab. It is extremely easy to get not only contaminate a swab, but get the wrong result by using the wrong area or not being reproducible. You need to put your swab people through very extensive swab training, which should take a minimum of half a day. And then they also need to then continually test themselves afterwards. There are techniques available to do that and to be able to test yourself to prove that you can get the right result, not only once, but every single time. Because unless you do, you are only, as we said earlier, you are only testing what is on the swab. You are not testing what's on the plant. So you have to make sure that the swab is representative. And you can only do that. And it's not just a case of following a procedure. It's a case of keep practicing and testing yourself until you are happy that what you've got is correct.
So you would say it's not a case of rolling out the testing at the beginning of the training and then testing it with that analyst maybe once a year afterwards. We're talking about much more frequent and intensive training program that these people need to be put through.
I would say they do the first intensive one and then they self-test themselves every time before they go out and take swabs. And they make sure that when they do that, an example is 20 years ago, I'd taken thousands of swabs and I could have done it 100% every single time. And know I could do it 100% every single time. I didn't then need to take swabs for nearly 20 years, but then a couple of years ago, I had the need to go to Germany and take some swabs. I tested myself before I went, and I was terrible, and I would have produced failure after failure. And I spent nearly a whole day retraining and testing myself before I went to Germany to take those swabs, because I knew fully well that if I went out with the ability I had at that time, I would produce a load of swabs which were meaningless. So you need to make sure you're happy with your ability every time before you go out.
Yeah, I must say I hadn't appreciated what a dark art taking swabs was until I sat in one of your sessions, Brian, and it's not as easy as it looks. So thanks very much for that, Brian. We've now come to the end of our webinar. I hope that you found it useful, everybody, and we appreciate the questions that you've asked us. As we've not managed to answer all of the questions today, we'll take some time to respond to these separately outside of the webinar. The next webinar in this three-part series will be taking place in June, and the focus of this one will be on the analytical considerations for your cleaning validations, and I hope you'll be joining us for that. On behalf of the RSSL team, I'd like to thank you for listening today. Please don't hesitate to contact me. My contact details are on the slide deck, as you can see, and then I can take those questions and help you through those with Brian, and we can maybe support you on some of your project needs moving forward. Thanks very much for your time. Good afternoon.